Thank you very much. And I want to first thank Laboratory Medicine for the invitation and getting me here today. And then I also want to thank the Department of Laboratory Medicine for getting me here today. And if you're wondering what I mean, there's sort of one of those little graduate moments where 15 years ago, Margaret Kenny turned to me and said, point-of-care diagnostics, in a soft, quiet tone of voice, with the understanding that if you want to do something important, get involved in point-of-care diagnostics. So pretty much for the last 15 years, I've sort of been following that idea. And I think that was a stage when I think the idea was a little bit radical and disruptive and things like that. And what I am doing, like I think all good scientists, engineers, whatever it is I am, is sort of riding a wave. So I don't want to take credit for this, but I'm saying it's a wild ride, it's very exciting. And I have opinions from part of the wave looking around, and by the end you may have your tomatoes out and be throwing them, but hopefully I'll give you a bit of the philosophy of what we're trying to do as widget makers. And then I'd love to get feedback from you about what you think about some of those ideas, and then I'll tell you a bit about this project that we've been working on, funded by the Gates Foundation. So I love technology, and I think the 20th century was an era of fabulous development of technology and diagnostics. If you want to be sick, this is a great place to be sick. People can find out what's wrong with you very, very effectively. We have big hospital centers and centralized labs. We do diagnostics that require trained personnel like you folks. You often consume large volumes of reagents and samples. Often you have large, expensive equipment. MR machines are wonderful diagnostic machines, but I wouldn't want to take one home. Some results are quick, some are a little slower. The things like microbiology can be a pretty slow process for a couple of days in certain cases. If you're a patient, you may have to go drive through traffic jams at 5 o'clock around here, sit in a waiting room for a while. And there's certain things you can't really do very effectively with these diagnostics, particularly with respect to things like outpatient clinical research and clinical trials. That's the 20th century. And I think of the 20th century as being pretty much where I was when I was in graduate school and I wanted to do computing. And that meant you took your stack of cards to the computing center and they did your computing and you got the results. Today, we have a somewhat different paradigm, and that's sort of the distributed idea, the idea that we are now, hopefully, moving to the point where we can take what we've done for computing and move it into some sort of aspects of medical diagnostics. These are three simple examples: the iStat, a pregnancy test kit, and a glucose meter. Three different sets of types of instruments. And frankly, the iStat was pretty much the inspiration. Margaret Kenny, I think at that time, was actually doing clinical trials of the iStat here at UW. So I said, that's pretty cool. What else could you do with that? And that's pretty much where we went. So when we think of this century, we think of a lot of testing occurring at point of care, ER, doctor's office, workplace, home, that a lot of people could operate the instruments themselves, that we could use small samples of reagents, and therefore we could probably get them ourselves rather than requiring a phlebotomist. We'd like low-cost disposables as a paradigm from the diagnostics industry, maybe an inexpensive reader, sampled result in under 10 minutes perhaps, rapid data connectivity into, and we'll talk about that in a second, and we think it'll enable all sorts of things. One, better quality of life and improved health care for individual patients, plus new types of theranostics, closed-loop therapies you couldn't have done otherwise, new types of ways in which you can do clinical trials and field medicine. And we'll broadly go back to that during the rest of the talk. And we've been, if you've heard Professor Kim talk, you've heard the phrase distributed diagnosis in home health care. This is sort of a buzzword, but it's one that describes what we've been thinking about for the domestic, let's call it the developed world market. The idea that you have some sort of an instrument, and 10, 12 years ago we thought it was a laptop computer, and it would talk to audio-video links and assay modules for chemistry, which is where I spend most of my time, or wearable and implanted instrumentation. And today we think it'll talk to the internet, to a medical database, essentially your data someplace. I don't want to say where that resides and how encrypted it is, but that's got to be part of it to identify trends. And then some combination of new data plus the trends goes out to primary care providers, specialists, clinics and hospitals if that's what's necessary, and if it's really necessary to the ER, this guy's coming, the ambulance is on its way, and he's got this. And even clinical researchers could get access into data they could not possibly get today if you could send it home. Now, we thought it was a laptop. Today, I would argue that the right tool is the one sitting on my belt, which is a cell phone. The cell phone today is pretty much what the laptop was 10 to 15 years ago. It's an incredibly versatile tool. It comes with all sorts of cool things like cameras, so I can take image data and move it anywhere on the planet in a few minutes. Mechanical actuators, all sorts of tools that are almost a laboratory diagnostic instrument. The part that's missing is the chemistry. So if you want to think about what I've been trying to do for the last 15 years, it's trying to figure out how to get the chemistry into this thing, okay? Into it and out of it, okay? In fact, you can go to sleep now. That's the main point. Who would use this? Well, right now, this sort of stuff, and since the iStat was around, is hospital inpatients with existing devices. But clinical trials are something you could think about, monitoring hospital outpatients. Chronic conditions are certainly something that's applicable for this kind of testing, to look for trends when people aren't in the clinic. Monitoring drug treatment, both chronic and critical drug treatment, is a really big opportunity from my point of view in improving healthcare. Transient conditions like pregnancy, early warning of emergent conditions in people who are at risk would be a really good way to go. And finally, something that we've found is a very good starting point for a lot of this is anticipating, preventing, and managing outbreaks of infectious disease. And epidemiology is one aspect of it, but certainly better treatment of patients is the primary focus. Now, for about the last 15 years, we started out with a very simple idea was, well, let's just work with small samples, and that should be just the same as big samples, but smaller. And what we discovered was the physics of fluids in small spaces was actually a bit different. So we now call this microfluidics as a general term. It's sort of another nice buzzword. What it really means is you've got a pipe with a diameter less than 1 millimeter and probably bigger than 1 micron, somewheres. That's all it means. However, people have used silicon microfabrication and these sorts of techniques used for making microcircuitry to make these devices. So you have the potential to automate complex procedures. You're compatible with small sample volumes. You can pack a lot of tests into small spaces. You can do a lot of parallel processing on a single sample or multiple samples in a single space. In theory, you can integrate lots of types of devices, pumps, detection units, processing components on the same thing. You don't have a lot of waste because you're starting out with small volumes of everything. In theory, you get better reproducibility. Why? Because you eliminate hand movement steps. Now, we can do that today in big labs with robots, but in fact, you could do even better than some of the robots because you can eliminate a lot of liquid transfer errors in these things, which is where a lot of the error bars come in most of analytical chemistry as we do it today. You have the potential for mass fabrication, and that should be extremely valuable if the following condition is met, which is you have low cost for the mass fabricated parts, and that's the goal. And a lot of what we do these days starts with the issue of low cost, as you'll see again. Now, we learned a lot about fluidics, and I'd say one of the interesting parts of the trip, as it were, was that those of us who were trained as chemists and biochemists, as I was, didn't learn much about mass transport. And it turns out what the chemical engineers knew was really useful stuff, particularly at small volumes. And we learned a lot about mass transport and how fluid moves in ducts, and were able to, it was like having a second career learning about this stuff over the last 15 years. And over that period, we developed lots of widgets and devices. It's sort of the list on the left is sort of like the equivalent of the transistor, the resistor, the capacitor for making electronic circuits. Some of these were developed in my lab at UW or colleagues' labs at UW. Some of them were developed at Micronics, which is a startup company that was founded based on the IP that came out of some of our funded projects from DARPA and venture capital people. I have a financial interest in Micronics, so I have to state that for the record so that you recognize that you can always ask me hard questions about this later, but their name will pop up again during the course of the afternoon. So lots of different types of components. This one over here we called an H filter, which is essentially an extraction circuit with no moving parts, which allows you to extract solutes from, let's say, a blood sample flowing in over here and flowing out there by running a buffer sample in here, and only small molecules or things that diffuse across the midline get into this channel over here, but you don't need to have a filter in the way, so it never clogs. All you have to do is keep running the fluids in at the right rates, and this thing will run forever. That was sort of our seminal idea, and from that a whole bunch of other things came. The idea that we could use the same device for a sensor technology by running two fluids into the same channel where the dimensions are on the order of microns to a millimeter, and looking at things like chemical changes like pH indicators to give us a pH meter with no moving parts, which is what that thing over there is, which is kind of exciting. The angle of the line between the red and the yellow is the dial indicator that tells you what the pH is. And from that we generated immunoassays in very small spaces that were extremely fast, on the order of 15 seconds actually for some of these assays, which really began to open our eyes to the things we could do in small spaces. So for the last 10 to 15 years, we've been trying to exploit things we could do uniquely well in small volumes as opposed to just scaling down bigger things. And we've had a lot of fun with that, and a lot of that patent list is from those sorts of ideas. We also, in collaboration with Micronics, in the earliest stages, said, well, silicon's not so good, what about plastics? Plastics are cheaper. And they went out and found a technology which basically is the same technology used to make bowling trophies, for using CO2 laser cutters to cut parts out of plastic sheets, which have on them essentially double sticky Scotch tape. You peel off the backing, and you cut out patterns, and you stack them up. And now you can build complicated fluidic circuits out of multiple layers that you can stack up. And when you're done, you can make devices that look kind of like this one that Micronics made about almost 10 years ago now, which is essentially the guts of a flow cytometry system. It's got about 14 layers in it, and this one actually happens to have a couple of silicon parts in it too. But you can make complex, multi-material things that are the fluidic equivalent of electronic circuits, but for moving chemistry around. And that is, again, about credit card size, which is a format we like, because you can pick it up and move it around. What we do today in the lab is we design things in CAD programs, like the structure in the upper left-hand corner is a bunch of different layers using AutoCAD or similar CAD programs. And then you basically hook up your computer to this box on the right, which is a CO2 laser cutter, and you hit print. And it will print out each layer individually. And you can make 10 or 15 or 20 of these devices per sheet of 11 by 13 plastic paper. And when you're done, you have a bunch of little bits of plastic, which are different parts of the circuit, and then literally you will stack them up. This particular one here is something we're using for the salivary diagnostics program I won't be talking about much, but you stack them up in sections. That's a PDMS part on the right over there. There's a gold slide is there, and so you can have mixing and matching different parts, and when you're done, you end up with a microfluidic circuit that's ready to roll. And the beauty is you can go from that CAD design to a finished part in about 4 hours. So rather than doing the silicon microfab thing that may take days or weeks to get something done, we can actually do this in hours. So it's really good for graduate training. We really like that aspect of it a lot. So we have what I'll say is laboratory goals of a sort. One, disposable polymeric laminate cartridges that use only a couple of drops of a biological fluid. I've listed some of them there, and they could include blood, urine, tears, saliva, or feces. Cost less than perhaps $5, so something you'd put in your medicine cabinet. Contain all the chemistry you need for multiple quantitative bioassays. And we would like to go quantitative, although there are obviously cases where we don't need it. You want to be able to put them in the glove compartment of your car for a year. So that means storage of reagents is going to be not like you put it in the -80 freezer or even the -20 or even in the regular 40 or 4 degrees C. We really want to go with storage of reagents, and that's a challenge, but not impossible. We want it really simple, so your 5-year-old could do it. We'd like the results in under 5 minutes. One simple reason is that's about how long I tend to get with my primary care physician, and I'd like to be able to think that from the time I walk in and say I feel lousy to the time I leave, we will actually know why I feel lousy, or at least from a set of things have ruled a few out. Rather than, well, go down, take some blood, and I'll call you in a week, which is more typically what happens, I think, for most Americans in good medical care. We'd like to shorten that substantially, and as you can see, that has big advantages in the developing world as well. We'd like, quote, lab quality analysis, meaning we don't just want a dipstick lateral flow immunoassay test where you get a yes-no answer. We'd like something reasonably high quality, and if necessary, a reader. If the reader is required to get the numbers out, we'd certainly be willing to do that, but we'd love to avoid it if we could. We're also these days thinking globally, and it's partially because I finally listened to one of my students and went over and talked to people at PATH, and sort of the blinders came off and the scale widened, and I realized there are things we could do here that are actually useful. Now, this isn't the only part of the world where you can do technological fixes. I invite you to go to sub-Saharan Africa and look at cell phone reception, and look at the fact that you can get better cell phone reception in parts of sub-Saharan Africa by far that are far away from cities than you can sitting here in this room. It's truly remarkable how that particular technology, for reasons that have nothing to do with medicine, has exploded across developing world areas. That's a really important sign, and if you can use that cell phone again as the mechanism for getting the data in and out, you've got a really big advantage. So, like I said, look to the consumer technologies for the way to get the data in and out there. Obviously, problems of health in the developing world can rapidly become problems for all of us. It's not like it's their problem or ours, it's all our problem these days. But if you're going to try to fix a problem in the developing world, it has to be an inexpensive solution. It can't be the MRI in every hut in the village. You can't do that. You have to come up with something that's inexpensive. So we try to focus the technologies on things that are inherently inexpensive and work backwards and outwards to how well we can make them work these days, which is certainly not how I was trained to operate as a sort of chemist in the old days. So obviously there are loads of things that are different about developing world environments, and I apologize for suggesting things you probably know very well. This is in India, this is an AIMS hospital. This is a really nice place to get sick in Delhi. Okay, it's a really good hospital. But an hour's drive away, you've got regional clinics that are not quite so wonderful, and this is pictures taken by our colleagues in PATH when they were doing one of our, as you see, user needs assessments. The lab conditions are not as good as you'd like to see in many cases. There's not a lot of storage. This is the lab bench over here, and this is pretty good because they've got power sometimes. They actually have battery backup with an inverter, so you can see a rack of batteries on the floor. They have a centrifuge and a microscope. That's pretty good, but that's about it. Okay, so the conditions tend to be fairly crude in most of these places, and some of them are much worse than this by far. There's also the lab supply business. How do you get supplies there? And typically this is a good example of a supply truck, and PATH has done a beautiful job in maintaining cold chain for vaccines, but you can't rely on every possible kind of supply being maintained in the cold, particularly when there's no points as when it gets there. There's no way to keep it cold until it's used 6 months later anyway. So the conditions are different, the technologies have to be different for what you're going to do. So technical issues include not everything's appropriate for the developing world. You can't just port something over there, and this has been a problem I think a lot of Western philanthropic organizations have run into, which is they simply take something that works here in your labs and try to move it directly to some place where it can't possibly function. Obviously there's some diseases which don't exist here, so there is no technology here in this developed world environment for solving problems in the developing world. Sometimes you can't even get samples for these diseases, and there's not much infrastructure in the developing world for running the tests once you've got them there. There are lots of non-technical issues. Poverty is a very simple fact, and it means that people don't have a lot of money, and what money they've got is often coming from either philanthropic sources or some sort of foreign aid program, which means it's very politically controlled. The burden of disease may be very high in many areas, far higher here, such that even the clinicians have got the diseases they're trying to study, which is a problem. Production facilities, if you want to make something, are not always available for qualified supplies. You may have a political infrastructure that's either unstable or corrupt, and as regulatory and IP issues also come up there, and they're different from the ones here. And finally, you have occasional wars that just take whatever plan you had, tear it up and throw it out the window. And the situation in Kenya is one that we're sort of acutely feeling right now, as you'll see in a couple of minutes. We've got 3 diagnostics projects running in the group, and I'm not going to talk about the enteric pathogens one, which is led by PATH. It's very exciting and it's very cool, and it's looking for the first time at a really rapid diagnosis of what's causing an enteric pathogen disease, meaning 20, 30 minutes to a diagnosis rather than 2 or 3 days. So it can actually make a difference for the patient. That's pretty exciting. It's funded by NIAID. We're also doing a program funded by NIDCR and a few other organizations on looking at saliva as a source for small molecules as a window into what's going on in the body, primarily for looking at therapeutic drugs and titrating levels of therapeutic drugs, which is another area which I'm not going to talk about. What I will talk about is the third one down here, the Bill and Melinda Gates Foundation Grand Challenges in Global Health. So this project we know affectionately as the Diagnostics box or the DX box, which is a pun if you're from New York, because that's what the game thing, it's called DX box, but that's just using the New York accent that I don't always use. It's supported by the Bill and Melinda Gates Foundation. This has been a very exciting and somewhat nerve-wracking experience. They have a lot of money, huge amount of support behind it, but it is kind of like being strapped to a freight train at the front of it, and there's a lot of energy behind you, but it's a thrilling ride. The focus is what was called Grand Challenge 14, and they identified the grand challenges. It was technology that assess individuals for multiple conditions or pathogens at the point of care. It's a 5-year project. I'm the PI of the project, so UW is sort of the central one, but trust me, most of that money goes right through my hands and out, as you'll see. The aim is to produce a system for rapid diagnosis of diseases for use in the developing world. It's a platform development issue, and we picked a particular platform, but we'll show you that in a second. It's a team, and the team is, like I said, led by me, but it's got Pat Staten in bioengineering, who does what I'll call the nanotechnology and systems development for improving on some of the technologies. Most of the rest of it we have begged and borrowed from existing unpatented technologies as much as possible to get it out there without having to worry about patent fights with other people, which is a tactic in terms of getting a device developed. Nanogen is very heavily involved in nucleic acid development, and they've been immensely helpful. These are the people formerly Epic Biosciences up in Bothell. They're really good. Fred Bottrell is the PI at Micronics, the startup company I mentioned to you earlier, and they subcontract to a company in Australia called Invitech, which is doing the instrument design and build. Micronics is coordinating card integration into the system. And then Path, with Gonzalo Domingo in the lead, is doing several different functions. Assay validation, meaning they're making sure the assays that we're going to try are the right ones in the first place, which means checking out a wide range of reagents and tests on as many samples as we can get our mitts on. UNA is user needs assessment, which is essentially the market validation, and then the systems testing. And the reason we have user needs assessment and systems testing is the aim is not just to do a research project, but it's an R&D project where the end point should be a functional prototype that can do these tests and is ready to be tested in the developing world. So it's not a very simple academic project by any means. It's a lot of people. I'm not asking you to look at the people, but there are many of us in many places who are working on this all the time. It's a busy thing. We meet teleconferences weekly, and every month we get as many of these people together in one room as we can. We don't move to Australia. They have to video conference in. This is the timeline roughly. We're at about the midpoint in a 5-year program. So we've done a lot of the things on the bottom already. We're sort of at integrating assays, which is a very, very interesting, important part, onto that plastic laminate technologies. The instruments being developed, and we're at this point doing one of the user needs assessments, and then we'll, in the 5th year, be doing things like optimization for 2nd generation tests. And you'll probably ask by the end, what are those 2nd generation tests? So what is it we're making? This is the wooden block mock-up. We actually had one of these made so they could take it to the developing world and show this to clinicians and say, if you had something like this in your office, where would you put it? How much power would it need? How could you use this? To give them a real feel of what's going on. The little plastic white things are the disposable cartridges, injection molded. You get an idea by someone's car keys how big the thing would be. Notice no wires. It's meant to be battery powered. So it's a platform for differential diagnosis of disease states. The aim was to have it detect about 6 pathogens by at least 2 methods. Not just one method, but 2 methods. And the 2 methods are 1, PCR for amplification of nucleic acids. It was meant to do rapid point-of-care PCR within the instrument on a disposable. Okay? The second is immunoassays for looking at either antigens or antibodies. So, and we'll talk about that more in a minute. Turnaround time of 15 minutes, I'd say we're aiming for about 20 right now is the best turnaround time for the whole thing. That means 1 sample in, 20 minutes later, you've got PCR done and immunoassays done, and you've got a readout. What is the readout? Most of the people at the user end want a yes-no answer. It's malaria, not dengue. Okay? There's an 82% probability. They don't want that gray stuff. And so we're aiming this essentially to dumb down the answers to a point where you can give what is either a definitive answer or I don't know. Okay? And that's a really critical issue about the user needs. Different users have different interests. The disposal should have all the reagents. The box should remain dry. All the stuff, all the sample gets retained in the cartridge. Solvents, meaning buffers, stay on there, as well as all the reagents. The instrument will basically use pneumatics to move the liquids around, but the liquids will never spill back into the machine optimally. On-card validation. Some on-card and some will be lot validation for these things. We want it small. It's about the size, as you can see, of about a shoebox. Rugged, battery-powered instrument, so you don't need the mains power. It needs to be very simple, so people can operate it under stress and bad conditions. And people who have very little training. The cost has to be low. How low? We're thinking a couple $100 retail cost for the box itself, okay? And then sub-dollar per cartridge for the tests. Okay? Low maintenance is an extremely important component. Where do these ideas come from? Well, partially they were our starting ideas of where we wanted to go that was validated by the grand challenge itself. Part of it comes from these user needs assessments. And we started in India in 2006. Completed one in Brazil in 2007, and there's one in Kenya that is, as you can imagine why, pending, because we got a bunch of data before the election. And then all the people are pretty much hiding at this point, both in Nairobi as well in Kisumu, where they did a lot of the data gathering. So this is sort of one of these examples of the real tragedies of working in some of these areas. Is you can plan anything you want, but reality will sometimes intervene. So we're hoping that we'll be able to complete the user needs assessment because it got most of the data out. But it's certainly by no means an optimal situation at the moment. Why Kenya? Because it was considered one of the most stable democracies in Africa. So who are the end users? We're thinking about the ones in white, okay? The developing world users. Those are the ones for whom this instrument is being designed by the Gates Foundation. I confess to you, though, that the corporations wouldn't be nearly so interested if it weren't for the fact that there are potential users in the developed world. So it's, in a sense, a dual-use platform. It would have different end users in the developed world, but the hope is that by establishing a technology and showing that it works in the developing world, we can eventually get interest in it back here as well. So what are our targets? Well, we picked, with PATH and their decisions, a set of targets that we get from one sample, in this case that's blood, ideally information about the following 6. They are dengue, measles, influenza, malaria, typhoid, and rickettsial diseases. The specific analytes are listed on the right-hand side. What's interesting about these is 3 of them are RNA viruses, 1 is a eukaryotic parasite, and 2 of them are bacteria. So you could say, well, I could use RNA or DNA. The common theme, however, of course, is RNA that should be available through all of them. So in fact, we've focused in on RNA as the sample, which is, as you know, rather volatile, doesn't last very long in most samples, so you have to be careful about how you get as fresh samples as possible or preserve them very effectively. In addition, we're looking at, and for malaria, we're looking at 2 antigens, one for specifically to Plasmodium falciparum and one that's a pan-Plasmodium antigen directly. And then at lower concentrations, after we've diluted things, we're going to look at IgM for all the rest and look at IgM. So we're doing, ideally, these things in parallel. We're not convinced that all of these assays will work equally well in a fingerstick sample of blood. Most infamously is influenza, and there's a long set of discussions we can have after that. It's not clear that we made exactly all the right choices in all the analytes, and that's part of what we're learning as we go along. Why IgM and nucleic acids? Well, this is a set of data from a study done not by us, clinical microbiology in 2005, on dengue patients who came down with rapid fevers after they came back to Sweden. And what it shows is, and this print's kind of small, this is the levels of nucleic acid over here is this curve. Nucleic acid titers are quite high immediately, and they drop off to pretty much nothing by day 5. On the other hand, in the meantime, the IgM titers against dengue are going up. So if you have the ability to look at both IgM and viral RNA in this case, you should be able to lock in the fact that you really got dengue at multiple stages of the disease. This is one of the major reasons we really insist on doing it both ways at once. It's kind of an exciting opportunity. So we think it could be useful at various stages in the disease process, from early on when you've got viremia, ideally, and you can catch things well, as well as after the viremia or bacteremia drops off, and you've got IgM titers and symptoms. We're not at the moment thinking of looking at the IgG. In fact, we have to get rid of the IgG for most of our assays, because that only tells you that someone had the disease 6 months ago or 2 years ago. And in the developing world, in regions where, for example, malaria is endemic, everybody's got anti-malarial IgG. It doesn't help you to know that. It doesn't tell you why the person's sick now. So we actually have to differentiate them quite well. What are we going to do with this thing? Well, we're thinking it's as simple as fingerstick for a couple of drops of blood, add blood to the disposable card, insert it into reader, and then you'll do some sample conditioning. Basically, we're going to split the samples. And one line will go into the immunoassays, the other line will go into nucleic acid assays. On the immunoassay side on the left, we'll do an absorbance imaging of spots on a membrane, as you'll see. We'll analyze the images, and from that we're going to get an answer from that line. We'll be similarly doing nucleic acid assays. We'll talk about both these lines in a couple of minutes and give you an idea what we're doing. You'll get fluorescence values on this side, and then you'll combine that information to determine the presence of a pathogen. And there's going to be a logic path that we're going to be working out, and this is to be determined, but it's going to be part of years 4 and 5 once we've actually got the numbers on this system. And then report the presence or absence of the pathogens, and it's not always single pathogen that you find in the particular sample, with obviously some level of confidence attached to it. As I said, the team members are doing very different things. Pat Staten's group is really developing some very exciting technologies which we want to use for what we call smart chromatography and target capture and concentration and detection. This typically involves modifying molecules and nanoparticles and is pretty much out of the scope of what I can talk about today. But it's new technologies very specific to the use of these kinds of diagnostic devices. And it's proven to be really exciting even for things like dry reagent storage, which is obviously a technological challenge to keep antibodies or enzymes alive indefinitely at 40 degrees C. The folks at Nanogen, and I don't want you to look at all the numbers, these numbers are just to tell you something simple. What they have been doing is they have been coming up with primers and probes for all the pathogens and then optimizing the primer and probe sets to not only give good sensitivity but also to be selective against all the other possible pathogens that are out there. So we're going for discrimination as well, and there's not only getting the optimal primers and probes, but we're biplexing every single one of the tests. So if we have 6 pathogens, we have 6 separate channels for the PCR. Each one of them will do one particular pathogen and an internal control. In this case, it's a housekeeping gene that's present in red and white blood cells so that we know that a blood sample actually got there. So if we have a negative, it isn't because no blood got to that particular channel. We have to validate all the fluid movements throughout the device because it's sealed in a box. The simplest way to do it is to biplex these assays with a second wavelength with a separate probe on that particular analyte. And Nanogen's been handling all that and gone through about 6 generations in order to optimize for the particular pathogens and the clinical samples we've been finding. Interesting problem. How many people in the room here have done PCR? Okay, I'm getting about 50% of the hands who've actually done PCR. But for most of us, the most important part of doing PCR is that big power cord we plug in the wall, because they're heating and cooling and heating and cooling and heating and cooling. So the heating and cooling rate is one of the limitations, and the amount of power you need to heat and cool is very tricky. There are different ways for doing it, but typically it's very power intensive. But we started with the condition that we're going to use basically a laptop battery to run this thing. And there was no way that something that required a thick power cord was going to work. So the original idea was, well, we'll do this with essentially a Peltier heating element, which is one of the ways people do these things, and put it on a card. And you have a container, and you ramp the temperature of that container up and down and up and up and down, maybe 40 times, okay, as the process of PCR reaction. So the temperature is, the container is what's cycle, the container temperature. You know, it works, and it's actually very fast on card. We got down to, I think it was sort of 8 minutes for 40-odd cycles for a PCR reaction. So we're doing really well. And we thought, that's great, but the power cord was still there. And we had arguments about power, and the folks at Invitech who were building the box got back to them, and a long delay ensued, and concluded that the Peltier element just uses too much power for our purposes, which is to run all day without requiring a recharge or a separate battery. So we switched the way we're doing it now, and this card on the right-hand side sort of gives you some idea that there are multiple wells. Down here is the sample processing. Here there are multiple wells that will actually be held at different temperatures. So now we're moving the sample back and forth through containers that are held at fixed temperatures above ambient. Good news is they're all fairly high. So even if you've got a 40°C room, you're probably okay for PCR because everything is above ambient. So you heat the containers and you move the samples back and forth. And that takes very, very little power once you've heated up the blocks. And as you'll see in a second, it's good. So the power is used about as efficiently as you possibly can do it, and that was a critical issue for this. So here's the general idea. You've got 4 blocks, maybe hunks of copper. You walk in, you plug it, you turn on the machine, and it's cold. In fact, we'll have 6 parallel 4-zone channels, but this is just looking at one of these things. Then you bring in your card, which is of course cold, and put it on top of the blocks. The next thing you do is you turn up the temperature and you warm them up. And you warm them up to 4 temperatures, 55, 95, 56, and roughly 55. Those are easy to do, and once you've heated them up to temperature, very, very little power gets used to keeping them at temperature in the box. So you put your sample in at 55 and you run the reverse transcriptase reaction at 55. You then push it with a small pneumatic push into the next chamber, which gets you to 95. So you inactivate the reverse transcriptase enzyme, denature DNA, then to a 56 anneal PCR primers channel, and then back to 95, and then back again to 56. And you do that maybe 40 times. Okay, and that's but you're shuttling. And each time, all you're doing is pushing the liquid a tiny distance, which takes negligible power. At the end, we run it to a block that starts out at 55. We look at 2 different wavelengths, one for the sample, one for the internal control, and then ramp that to 95C, and we detect at those same 2 wavelengths. So we get 2 points and 2 points, and we're done. That's all it takes. It's a very simple way to go, and it changes the power usage enormously. This is just a picture from about a year ago showing that you can actually get the temperatures changing appropriately with liquid shuttling back and forth on the card, just to show the temperatures move. They don't stay exactly constant because when you move the liquid in, the liquid's cold and it has to heat up, so the temperature drops for a little bit and then comes back up. Does the opposite thing when you go the other way. It works. This is an example at 10,000 copies for the malarial detection, which was something we did almost a year ago now. This was with a 6-well card, 40 cycles in under 20 minutes. It was a little bit longer than it was on the Peltier element. We're still trying to optimize and keep shrinking the time, and it's down under 20 at this point. This was at 10,000 copies, and these little bands that show up down here are the indication that we actually got success in the amplification. We're now at substantially less than 1,000 copies detection, and the target is to get 100 copies detection per channel. Okay? If you think you split it 6 ways, that's actually an interesting thing that you have to do. So we're not talking about 1 copy detection. The assays, as run by Nanogen, are single copy detection absolutely cleanly. Okay? They're very, very good at single copy, but we degrade by putting it on the card itself. I want to briefly then tell you about the immunoassay side, which is where I spend most of my time. And in fact, there really are 2 immunoassay efforts going on, one at Micronics, one in our lab, for a bunch of really good reasons. The Micronics one is on the right, and basically it's a flat capture film onto which have been deposited capture antibodies or antigens in the case of the IgM detection. And then we go through a series of washes and rinses, and the net result is they are then amplified using the horseradish peroxidase TMB assay. I don't know how many people use that in lab medicine, but it's used a lot in histology to use the TMB dark blue precipitate reaction as a way to get things. It's the same reaction that you use just for getting blue color in ELISA, but with a precipitant that causes the blue stuff to stay fixed at a site. And in fact, we're using the same thing on our side with a different idea. This is a flat membrane and things wash back and forth. On ours, we're actually pumping liquids through a porous nitrocellulose membrane and immobilizing things on that, and then doing the same amplification reaction on the porous membrane. It gives us about 250 times the surface area, but some additional fouling issues that are fine. So very easy to detect either with the TMB reaction or another approach where we use gold labeled antibodies, and we'll talk about that in a minute. So I'm going to tell you mainly about the left side and where we're going and what that is. Really simple idea, which should be very, very obvious to all of you, and I apologize for belaboring the obvious, is we have a membrane, in this case nitrocellulose, about 150 microns thick, and then we can flow things through it. And what we're basically building is what we call a full stack immunoassay with a capture antibody on the bottom, like this. So we put the capture antibodies on, and then we add a blocking solution, and we dry it and store it. And ideally, we hope to store these for a year or 2. Then we add a sample, which has in it the antigen. The antigen is captured on the surface. Once we've rinsed through the sample, we add the labeled antibody, which can be either horseradish peroxidase or HRP or a gold label one. If it's gold, all we do is rinse the excess and read, and we're done. If we want amplification, we then add a chromogenic substrate for HRP, like tetramethylbenzidine or TMB. That converts and precipitates, and we get a dark blue precipitate all over the sample membrane itself. And ideally, it's localized, so we get a spot where we put the right capture material on the membrane itself. So we can either look at the rate of formation of the blue stuff or how much there is after a fixed period of time after a rinse, in either case. This helps. Microfluidics really speeds things up. If you look at conventional ELISA assays, we tend to think of these as taking hours to complete because there are lots of incubation times and you have a limited amount of surface area and a relatively large volume. That's fine, but what we're doing actually can speed things up to less than 10 minutes. And we've had a lot of luck going to those sorts of speeds for these assays. This is just an example, again, of a flow-through membrane, and we'll have multiple spots, spotted as you'll see in a minute, on the membrane itself. This is just the last stage. This is developing a TMB spot. So this is a membrane sitting there, and Paolo Spichata-Mihalic is taking a pipette, and he's going to add the TMB solution, which sits on top of the membrane, which is being sucked through. And you can see over there the blue spots are showing up within a second of the arrival of the TMB. As it continues to flow through, they get darker and darker and clearer until by the time it's sucked all the way through and we have a dry membrane to look at, you get some very nice, highly resolved spots. Those are an array of 16. Actually, on the right-hand side, that's the PFHRP2 from the malarial sample. As I said, we've got 2 different versions. One is the TMB HRP on the left, and also the gold labeled antibody. The spots look comparably dark. We get about a factor of 2 or 3 better sensitivity with the TMB results so far, but that may not be necessary, and there are some problems with the TMB, which is we have to preserve horseradish peroxidase. So we got to keep an enzyme alive at 40C for a year. We're having remarkable successes. We were pretty despondent about that for about the first year, and then we got smarter and smarter, and it's looking fairly good now, both in our hands and in Pat Staten's hands, that we look like we might be able to do that, because it's really helpful to have the ability to do the amplification. So we're actually fairly optimistic about that right now. But we do have the gold as a backup, and the gold labeled secondaries are quite robust. We started out working on 96 well plate formats. This is a 96 well plate format vacuum manifold that we use to suck things through the membranes in order to get the spots. We've subsequently shrunk it to a smaller scale. It's about the size of a business card, a little bit smaller than a business card, in which we can array multiple spots. And we array them with an inkjet printer. Here's an example of some of the data taken from the 96 well plate format. This is the 2 antigens we're using for malaria, which is where we put most of our initial energy. And this is intensities of the spots. We call it a response to noise ratio over here, which is a normalized version of the darkness of the spots on the white background. Here's the PFHRP2 and the aldolase results. The noise level limit of detection down here is the red line. So we're down to about 6 nanograms per ml for the PFHRP2 and about 13 for the aldolase, which is, we think, perfectly adequate for the samples we're going to get. And these were done not in buffer, but these are actually spiked into fetal bovine serum. We've also done these things in pooled human serum samples and are having pretty good results right now. Comparable results, I should say. The spotting we do with a home-built inkjet printer. It's not exactly what you'd use to write your book report, but on the other hand, it's got the same general ideas of squirting small droplets down onto, in this case, a piece of paper, which is literally what we're using, except it's nitrocellulose. But we can control, drop by drop, how much stuff goes down and where we put it with a lot of precision. And I should say that the hope is that this is a technique which is readily convertible through commercial inkjet printing to something where we could print these things out for virtually nothing, okay, where it's just the cost of the tiny spot of the reagent. Because again, cost is a key issue. We wanted a manufacturing technology that was inherently, could be very inexpensive. This is just an example of some of the spots. This is to show you if we put down a single drop, we can space, in this case, 5 of them out through 800 microns. The spots can be darn small, so we can put a lot of reagent, a lot of different detection events in a very small space. So we can do not just 6 analytes, we could probably do 60 or 100 with redundancy across the membrane and easily fit it into a square centimeter, which is roughly our detection area with the camera at this point. This is just showing that there is a correspondence to the number of firings, and this is the TMB development. The droplets, as you can see, are down to 15 picoliters, so there's not a lot of sample there, which is why you can imagine having relatively inexpensive reagent use in these things. And again, we can biplex these things in all sorts of ways. This happens to be PFHRP2 on one side and aldolase, a different one on the other side. One is developed with the TMB, one is developed with the gold secondary antibodies simultaneously. So we can actually mix and match not only the specific secondary antibodies, but even the type of detection we're doing with these things. So there's a lot of versatility in this kind of system, even though we're flowing everything through a single common membrane. These are how we're doing right now is we've got very good control over the PFHRP2 and the aldolase, and we're doing quite well with rickettsia and measles. The IgM detections have a whole set of interesting problems I couldn't go into. It turns out it's a lot tougher to integrate multiple IgM detections using the version of the assay where you put the antigen on the surface and then detect IgM and then use a common secondary for all the IgMs. This is the I think it's indirect version we're using, and it's a little bit tricky. So far we got rickettsia and measles down and we're very comfortable. We're struggling with reagents and reagent suppliers. This turns out to be one of the biggest lessons for us is it's hard to find good reagents that you can use for an assay. We are worried a lot about storage. This is just showing that we have in fact done a fairly nice job of stealing from the people from lateral flow immunoassays, that we can use the same technology to store secondaries, labeled secondaries, and dry them down on fibrous pads, store them dry for long periods with good retention, and if we rinse liquids through, we can actually get almost all of the stuff out within a couple of seconds. So we can actually get a bolus of secondary antibody washing downstream when we want it in the fluidic system. So we built cards. This card was actually made by us in our lab, by Dean Stevens, and this had actually room for 2 conjugate pads. One was going to be for the secondary antibody, one for actually the TMB reagent. This one was actually tested, in this case, with the gold secondary antibody. And down here is the capture membrane on the card. And what I hope to show you in the next one is, again, this is a movie of, that's what it's going to look like at the end. At the beginning, you'll see here comes the dark purple gold labeled secondary antibody washing across the membrane. Not much will happen for about 60 seconds here as we slowly build up secondary, but what you're going to see is we'll get red spots where the stuff is. This is the Mark I test. We found all sorts of interesting problems with this one, so we've done about 3 redesigns since then, and then a major one I'll show you in the next one. But the bottom line is, if you wait long enough, and if you think about the entire time, it's sample in to reading the result with the camera in about 8.5 minutes. So we're very comfortable that we're going to be able to do this quite efficiently with at least 6 different analytes on card. We should be able to go to more if we can get all the reagents multiplexed. We're now going through a card that we had built at Micronics, which is substantially more sophisticated and does a lot of things with only pneumatic control. Last one actually had syringe pump control. And this is sort of the guts of the card, what it looks like, and this is what the actual card looks like. We've built in membranes to control airflow, so we can control how much liquid gets on there and meter just using airflow control. And the card itself is easy to manufacture and reasonably easy to use. This membrane over here is the piece of paper, so you can see we're only using one of 2 windows. Ultimately, we're going to put on the IgM test on the same card. That's something we hope to do during the course of the next few months. So where is this thing going? We have what's called an alpha prototype, which is targeted to be completed by about June 2008. The first of them should be done. The format is rotated a little bit. The sample now comes in from the side. We've picked out battery supplies and all the internal components. This is the idea of what the card will look like. There's a window over here for the nucleic acids and a window here for the immunoassay tests. Different sides of the card, different temperatures for the different parts of the card as well during their life cycle. And it will have an add-on printer, since many people in the developing world said they don't want that little readout, they don't want the cell phone, they want the piece of paper. That's what they carry to the patient. So we're spending a lot of time at the end users discovering what is the actual mode of readout they want to have. This is what it would look like with the drawer open. So you'd drop in this card, you'd hit the little green button, sucks it in, get started, and hopefully 22 minutes later you've got all the sample readouts going in there. As I said, we think we're moving along well. We think it's, as it currently stands, potentially very responsive to the developing world needs. One, it's going to be the first time you can bring PCR-based diagnostics to remote sites with very little power and low resource settings. You can get them there, but you can get them to the centralized hospitals now. This is really going to open things up a lot. 2, it's going to give you capabilities for quantitative and qualitative immunoassays, both. We may only use the qualitative numbers, but we could go more sensitive with it and more graded in the response. It's applicable actually not just to this panel, but a wide range of panels for diagnostic tests. All you have to do is modify the reagents on the card. It's a very simple thing of just reprinting the piece of paper, putting an indicator saying what's in those spots, and then the rest is done by the DX box itself. So potentially, we open it up to anything for which you can do an immunoassay or a nucleic acid test, or both. It's versatile. We both use fluorescence and absorbance measurements, so we got a fluorescence detection system, and we use exactly a cell phone camera, in fact, is the detector in there. That's how you can get the whole box to cost less than $150 or $200, because we're using commercial parts, stealing in every possible way from consumer products to make the thing work. The disposables should be inexpensive. We ultimately will end up manufacturing in the developing world. PATH has been very good at figuring out ways to do that, and Micronics, as the chief commercializing entity, has the job of finding people who can manufacture these things at cost. Now, eliminate may be too strong a word. Greatly reduces the need for highly trained technicians or clinicians or pristine working environments for getting these kinds of assays done. We hope it will fit just fine on that rather distressing looking laboratory bench that we showed you in rural India. Low cost, but still a sophisticated instrument capable of really good tests. No need for continuous electrical power, which is a critical requirement. And it's certainly integratable with informatics systems throughout the developing world. I don't think we've finalized what's going to be on the far side, but in the alpha, as you'll see in a second, there actually are a whole bunch of ports, from USB ports to FireWire to telephone cables. So there are lots of ways to get the data in and out of this machine. Just so that you think I'm not kidding, this is the shelves at Invitech in Australia who's doing the device build, and this is a picture taken on January 10th, 2008. All of the parts required for these things were on the shelves as of January 10th. They're busily building the first one of several they're going to build, and you can see their plans to make a bunch of these alphas. So it's real, it's very real to us, and we're eagerly anticipating the arrival in June of the first one. So I want to say first, thanks to a lot of people. Certainly my research group, which is a bunch of very busy people, both in the group and people who have been in the group, and lots of exciting collaborations with people listed along the bottom. and lots of funding sources that have made this possible. Going back way before the Bill and Melinda Gates Foundation to get us started. DARPA put a huge amount of money from the beginning. The venture capital people who eventually founded, from Senben Medical Ventures who founded Micronics were absolutely critical, and whole bunches of other people listed up there along the way, including NIH. Thank you, and I'll be happy to answer questions.
I love
Yes.
PCR, and it's a clever thing in terms of moving the fluid instead of ramping up things. But as you set up your intro to the talk, I was guessing in my head you were going to tell us that PCR was the wrong technology here. That you were going to tell us that you want to use an isothermal amplification.
Right.
So
And
why did you not go that way?
Okay, there's a very interesting question about it. The question was why PCR as opposed to an isothermal method, and why wasn't I going to say that? And the answer is there are other methods out there. The answer is interesting and commercial, and it has to do with the fact that if you were starting up a company and wanted to commercialize something and you had a choice between, say, taking something that was off commercial license because of the patents running out, as opposed to something where someone was going to extract a very large fee to use their technology, the idea was PCR went free and clear, I believe it was this year. That was the primary reason. Could we use the isothermal ones? They're not as generally as good, many of them, as PCR. PCR is still the standard for doing what you need to do. So it's better, and the question was, if we could make PCR work, we'd have a cheaper technology for the end users in the developing world, number one, and number two, we'd actually probably have better results. So I think there were two good reasons for going that route. Yes? Tim?
So so I was listening to you talk. It's great. You know, I'm a fan of your work. So one thing I was thinking of was environmental degradation or of the disposable part. So
Yep.
you talked about making it of plastic. Then I was also thinking about reagent uniformity. How do you make sure that, because you need, even though you have dry reagents on there, you still need some sort of, I don't know, clean water or whatever you're going to need. So
Yeah.
how do you make sure that
Okay. There are two questions there. One was about reagent uniformity, and the other one was about essentially the ecological impact of using plastic disposables. What I didn't show you in that picture when I showed the laboratory in rural India, the testing lab, which is a lovely place, and they do a really good job, but if you were to pan left and look 90 degrees to the left from the image of the front door, you would see their waste disposal site, which is basically an open yard with no fence, with needles, all sorts of contaminated blood samples, HIV samples, all sorts of stuff lying in a heap with dogs rummaging through. Okay, there are some really frightening examples of how medical waste is dealt with in the developing world. This is a general problem that goes way beyond this particular type of disposable. Needles, are certainly at the top of the list in terms of waste disposal problem. I can certainly see that adding to the pile of plastics, particularly plastics that contain a biological waste, is not the best way to go. And I think whoever thinks about distributing this is going to have to think about a disposal mechanism that works effectively. Ideally, all the hazardous waste will be contained within the disposable. So you could say we could come up with an efficient burn bag, but then someone's got to make sure the burn bag is burned. So, at the moment, I think all the parts should be combustible. I do not know about toxics from the burning at this point. I think that's going to be one of the engineering and environmental issues that's going to have to be dealt with this or any other technology. But you're right. There is going to be disposable. I don't think it's necessarily worse than some of the existing situations now that also need to be solved. Okay?
My only concern with that is if you have now this box that works, it's great that everyone can run on a battery, the number of tests will go up exponentially. So it's really not a one-to-one comparison. It's like you would increase the total number of tests. So it is kind of.
Yes, you could generate more waste because of this. But like I said, I think the problem is real, but I don't think it's unique to this particular class of things. As you increase therapy, the number of everything from pill wrappers to needles also goes up. So hopefully those will go up as fast as this, because the point of doing a diagnostic is not just to do a diagnostic, but to optimize therapy, which means you've got a therapeutic as well you need to deal with. The second question had to do with uniformity of reagents. And uniformity is a word I wouldn't use necessarily. I would say that, one, there's the issue of degradation of reagents. And over time, all these disposables will die. Okay? No matter how good you are, you put anything at 40 degrees C for a year and you will lose functionality. We're doing, to the extent possible, onboard controls to make sure that everything is working. For example, in the nucleic acid side, we're looking for the housekeeping gene. The level at which that signal shows up should tell us something about the PCR enzymes and how all those are working. And we're going to get an actual number. that will help us do that. On the immunoassay side, we want to run as many controls as we can on the card. We're going to be running, for example, we're not exactly sure which of the two we're going to do, but a blood protein as a positive control to guarantee that the blood got all the way through to the sample. And that will give us an indication that at least that particular antibody is okay. We're going to have to test off card the rate of decay of function of the separate antibodies, assuming that high temperature is the major issue. We also plan to do lot testing, meaning periodically you'll actually take a sample and apply one sample which has one of everything in it and apply it to one card and run the card in the device to make sure that that lot is still okay. So we're going to do a combination of different types of controls. They have to be controls, otherwise the numbers are meaningless because we'll just get a bunch of false negatives.